All right. Well, welcome to participants. Thank you for joining our Live and Learn faculty presentations hosted by the University of Oregon's Pre-College Summer Academy to Inspire Learning program, also commonly referred to as the SAIL program. I'm Laura Fernandez, Executive Director for SAIL, which serves middle and high school students in their pursuits of higher education. We are honored and excited today to be featuring 4 UO Physiology faculty members who will share what they do in their respective areas of research. So today joining us is Hans Dreyer, a muscle physiologist, and his area of research is to enhance muscle recovery and regeneration that leads to better physical mobility. We also have Andrew Levering, who is a cardiopulmonary heart and lung physiologist interested in environmental physiology, which includes altitude, heat, cold, space, and diving. I can't wait to hear more. Dr. Ian Greenhouse directs the Action Control Lab, and he will cover what we know about how our brains control our bodies and how this can be studied in laboratories. Dr. Andrew Karduna is a bioengineer whose research is focusing on the biomechanics and neurocontrol of the upper extremity. He studies the forces and movement and patterns of overhead workers, patients with orthopedic injuries, and baseball pitchers. If you have any questions at all during our presentation, please add those into the chat. We will happily answer those at the end of each presenter's speech. And we are very excited to welcome our faculty to our session. And Ian Greenhouse, you are our first. Welcome.
Well, thank you. Thanks very much, Laura. So let me go ahead and share my slides here. I just have a handful of slides I'd like to show. I'll make sure my chat is up as well. So go ahead and think about moving your left index finger, but don't move. So just think about it. All right, so you're thinking about it. Now, go ahead and actually move. So what's changing there? What's changing between the preparation when you're actually just thinking about a movement and when you physically move your body? And this is a question that motivates a lot of the research in my laboratory. We don't actually know. We don't know what determines sort of this threshold between just thinking about a movement and then actually moving. And we study this in the lab using a combination of different methods. One of the methods we use is a form of non-invasive brain stimulation called transcranial magnetic stimulation. So this is a tool where we'll have people sit down in the lab and we hold this plastic coil over someone's head, and we can deliver a pulse of electromagnetic energy that will actually cause a volley of activity inside the neurons in your brain, the cells in your brain, that actually goes down your spinal cord and can cause your muscles to twitch. And we can measure the twitches in your muscles. And we can do this when you're just thinking about moving or when you're actually engaged in movement, and try to better understand how the excitability in your nervous system, in your brain and your spinal cord changes while you're getting ready to move. So we want to know what's happening behind the scenes while you're getting ready to move. Another question that we look at in my laboratory is individual differences. So why are some people really quick to move? Why are some people really slow to move? And what are the differences that sort of determine this? So I just want to show you a short video. I hope that it's not too jumpy here. So this is a video of Fallon Sherrock, who's a dart thrower. And I just want to show you how amazing this individual is.
180, 140 I feel here, just to apply pressure. 180.
So I don't know if you could see the precision with which she was able to throw her darts. And here, I'm just going to zoom back just a little bit here. So these are how close together these darts can actually get on a dartboard. And she is extremely good at this. In fact, in the World Championships back in 2019 and 2020, she was able to do this back-to-back on multiple occasions. So why can somebody like this achieve this high level of precision, whereas somebody like me just can't even get close to this? Even her competitors who are well-practiced just like her, they can't even achieve this level of precision with their dart throwing. And one of the questions that we ask is, what individual differences look like in the brain? And we can use neuroimaging. We can use an MRI scanner to actually look at the anatomy of the brain, but also the chemistry in the brain. So we can actually look at how much of different chemicals there are in specific brain areas that we think might be important for determining these types of individual differences, meaning how good somebody is at a task like this. So we can measure the performance on a task, we can measure the chemistry in the brain, and we can try to link these things together. So these are the types of questions that we ask in my laboratory and how we approach it. So here's just a picture of the lab having fun with our non-invasive brain stimulators. So I'm happy to take some questions here. I wanted to keep it very, very short. I'll stop sharing my screen. I was warned there might be a bit of a latency between questions. Oh,
There
thank
might
you, Andy
be.
Lovering.
Let's see if we have any chat questions yet. You were pretty fast, Ian. I'm not sure we even have chat in there yet, but
Oh, okay.
Well, tell us a little bit more for a
Yeah,
minute.
so I'm happy to tell more. So one of the questions that we also study in the laboratory is, how do these things go wrong? So we look in clinical populations, including patients with movement disorders such as Parkinson's disease, and try to understand, in the case of their nervous systems, why is it that they can't do certain types of motor tasks and what goes wrong in their motor system? So we can look at the chemistry again, we can look at chemicals inside their brains and try to understand where in different brain areas these chemicals might go awry. So if they have too much of a chemical in one brain area or too little of a chemical in a brain area, how does that relate to their motor ability? And we can link all this together again to try to understand individual differences. And down the line, this might help treat patients who have dysfunction or have an inability to initiate movement or to control a movement. I'm happy to keep going since I don't see any chat questions yet.
Yeah.
So one of the findings from our recent work, which is kind of cool, is that we can look at how much of a chemical that is important for suppressing brain activity. So this is a chemical called gamma aminobutyric acid, or GABA. So this is a chemical that actually causes brain cells to become less active. And what we found was really surprising. We found that people who have more of this chemical in a particular brain area that's important for producing motor movements were actually faster to execute those movements. We thought the opposite would be true. We thought that people who have more of this chemical in their brains would actually be slower to execute the movements. But what we think is actually going on there is, if you have a race car for a motor system, so if you have a really fast motor system and you can make movements very quickly, you might need more of this chemical floating around in your brain to offset the excitability in your motor system, meaning that if you can execute a movement really, really fast, you want to have good brakes. So if you have a fast race car, you need good brakes on that race car. And that's sort of how we're thinking about the motor system is working, and what the role of this chemical might be in the nervous system.
So I have a question for
Yeah,
you, though, Ian. Sorry, I don't want to hog questions, but I am, this chemical, is this something that, if you don't have, like some people have more than others, I'm assuming by your dart person example, is that a chemical that you actually have, but it's pent up in your system and it's just not being released the way that some others is being released? Or
yeah, great question. So we don't know, but this chemical is something that's endogenous, meaning it's there to begin with. So gamma aminobutyric acid just exists in the brain. And like you said, different individuals have different amounts of this chemical in different brain areas. And it might be possible, we know that it's possible with different medicines and different pharmacology to actually cause people to have more of it or less of this chemical, and also cause more of it to be bound, meaning more of it to be used up in the brain. So we can actually encourage people, or encourage the brain rather, to use more of this chemical in specific brain areas. So this is something that we can also investigate in the lab down the line. We're hoping to maybe see if we can manipulate these things. And one of the ways that we might manipulate it is just through practice and training. So instead of giving people a drug, we can actually have people do a behavioral task over and over and over again and become very expert at that task, and see if maybe that chemical is changing, and maybe promote the changes in that chemical to make somebody as amazing a dart thrower as Fallon Sherrock. So there are ways that we can sort of manipulate these things. So I see a couple questions here. So the first question is it possible to create precision within an individual? So that's exactly the point I was making. So I think the idea is that we might be able to through practice regimes, or maybe through interventions of some kind, improve people's ability to do various tasks and maybe achieve a higher level of performance. So another question here is, are there any courses at the university that go more in depth into what I'm talking about? Does this chemical explain why some people have quick reaction times whereas others have slow reaction times? So this is exactly right. So I'm actually teaching a course right now called Motor Control, where we go into exactly these types of questions, and we study the motor system and do experiments in the labs. So one of the courses one of the labs that we do right now is actually a reaction time lab where we try to look at individual differences in how people do these tasks. And we can also do a lot of these activities remotely. One of the fun things we've been doing this quarter is capturing video of people moving on their phones, and then we can analyze that video to try to understand how people might move and how individuals move differently from one another. So yeah, we can do this in the lab. Unfortunately, we can't measure the chemical in the lab. We need an MRI machine to do that procedure. But exactly right, that this chemical, the amounts of these chemicals that are available might actually determine how quickly some people respond and how slowly other people respond.
Fantastic, Ian. I am very appreciative of what you're doing. This is I want to know what results in a year from now you come up with.
I do too. I want to know what we're going to have in a year from now, especially given the current climate.
Yes. Oh my goodness. Well, there may be some more questions that pop up. We have 3 more presenters. Should I pop to the next person now, or did you have anything else you wanted to add before we move on?
I will answer one question here that came from Dr. Lovering. He said, why don't I have an MRI machine in my lab? So MRI machines are very expensive, and the university has 2, which is great because we can actually use them. And I do have undergraduates who do research in my lab who have been trained to run the MRI scanner, which is a lot of fun. So this is a tool that is available to us. And because we're able to do our research on it, I'd like to make sure that students have the opportunity to control this giant machine and learn how to use it and operate it, which is, I think, a fun skill for undergraduates to acquire at the university. So yeah, just wanted to mention that.
Thank you. Well, thank you for your presentation. We'll be circling back probably with more questions, but Hans Dreyer is up next. Welcome, Hans.
Yeah, welcome. Thanks for having all of us. So let me just get a little bit situated here. I think I've already shared my screen. So continuing on the theme from Dr. Greenhouse, think of your knee. Don't move it, but just think about your knee. Okay, now extend your leg out like you're kicking a soccer ball. Okay, now think about those 2 conditions, not moving it and then moving it and always having pain. That kind of describes osteoarthritis. So that is kind of one of the things that we study, not necessarily the osteoarthritis itself, but how do we recover from this surgery that helps reduce the chronic and debilitating pain that occurs with osteoarthritis of the knee. So surgical remediation.
Sorry, but I think my video might still be pinned. Is it possible to transfer it over to Hans?
Oh. Can you guys see my screen?
There we go.
You guys can see mine now?
Yes.
Okay, yeah. I don't know that we need to start all that over again, but so here is a cartoon image basically of degenerative joint disease, osteoarthritis. So we can have here this normal cartilage in the white, and then here we have a breakdown of this cartilage. So this causes pain at rest when you're not moving your leg, and also when you are moving your leg. And this affects older adults. 60% of older adults over 65 have some level of osteoarthritis. Again, this is a cartoon. Here we have eroded cartilage, kind of points to. Here we have some bone spurs, causes some pain. Meniscus is some of the cartilage that helps maintain contact between this femur bone and this tibia. Again, this is the knee joint looking from the front. And here's exposed bone. So bone on bone contact, very, very, very painful. And this can, as you can imagine, will cause pain and even just difficulty sleeping. And so sleeping, I'll just put a plug in here, is a class that is taught at the University of Oregon by Dr. Levering, one of our fellow speakers. And it's becoming increasingly evident that sleep is of major importance to mental and physiologic health. And so one of the things that we don't study with this, but we would like to, is the effects of lack of sleep on this patient population. Here's an X-ray showing this degeneration. So here is a normal space between the femur bone and the tibia. Here's the fibula on the outside of the knee. And here's this bone-on-bone contact. Again, very, very painful at rest, and also very painful with any kind of emotion, say, for example, kicking a soccer ball or just getting up out of a chair, especially for older adults. Okay, here is a picture from the operating theater where total knee arthroplasty is occurring. And here you can see there's 2, this is an orthopedic surgeon and ostensibly some of his helpers, or another orthopedic surgeon. They're actually sewing up the after effects of having total knee replacement. And here is another X-ray of what a total knee replacement looks after it's been implanted down in the knee. So we have this is the femur, and this is the now artificial. You can think of this like the terminator joint here on the top, and the other terminator point. So metal on metal necessarily to take away that chronic bone on bone pain. And what my lab studies is how can we have a good outcome, in other words, preserve muscle mass after the surgery, versus what's typically happening, which we call, quote unquote, a bad outcome, which is typically after the surgery, they lose significant amounts of muscle mass. And this has issues with getting up out of a chair, walking across the street, again, thinking of older adults necessarily, just helping them maintain a healthy lifestyle, walking to the grocery store, going up and down stairs inside their house. And so we use essential amino acid supplementation versus placebo. These are double-blind, placebo-controlled clinical trials. I know it's a big set of words necessarily. It sounds all fancy. It's a lot of work. It's a lot of fun too. And we've had some success by just giving people essential amino acids versus those who are given placebo in the lead up to surgery and then for some period of time after surgery. So this is something that they take by mouth. We have a compounding pharmacy that will then make the amino acids. Amino acids, you can think of those as like the bricks, and a brick wall would be the protein. So this is a large component of what makes up muscle mass. And there's several different things about essential amino acids that are unique. Essential means you have to take them by mouth versus the non-essential, your body can make them. And also they have a fairly potent effect on muscle metabolism. They can stimulate or assist in the stimulation of muscle growth. And so we're using it to try to preserve more muscle after surgery versus placebo, which we've shown has regular muscle atrophy, which can compromise strength and balance, and again, getting up and down stairs. Here's an image of Kelsey Mosher, a student in the lab who's analyzing MRI images. I don't have an MRI in my lab either. Again, those are about $5 to $6 million. We now have two. It makes it very, very helpful for all the researchers on campus to do studies. Here is the total knee leg before surgery. So this is preoperative. Here's the control leg, so the opposite leg. So you have one leg that's going to have surgery on and the other leg's not going to have surgery on. And ostensibly the control leg is maybe doing a little bit more work because they're kind of limping a little bit. And here you can see the images at 2 weeks after surgery and then at 6 weeks after surgery. And individuals like Kelsey and many, many other undergraduates and graduate students in the lab will spend time analyzing the amount of muscle that is there versus before surgery. And then we can see, is the essential amino acid, our treatment, is doing a better job of maintaining this muscle mass versus a placebo. Here is Josh Mangum and Austin Hawker. Josh is looking through a microscope at biopsy samples of muscle. So this is a blown up image of muscle cells. So inside here, there's many thousands of muscle cells. We take a muscle biopsy from the vastus lateralis, and then we can analyze it. So not only does the MRI give us MRI gives us a whole quadriceps view of muscle, but then the biopsy will give us at the muscle cell level. And just to give you a little frame of reference, these muscle cells are about the diameter of a hair. So if you take your hair out of your head and look at it like on end, these are about that same size. And we have many, 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 many thousands, hundreds of thousands of those in our quadriceps, for example, which is the thigh muscle. Continuing right along. So then the goal here would be to look at TKA good outcomes versus TKA bad outcomes. And ostensibly, this is more prevalent in those who are having placebo versus those who are having our treatment. And again, we could do these studies in not just total knee patients, but other athletes having ACL surgery, for example. Again, cross sections of here's satellite cells. We can count these number of satellite cells. These are muscle stem-like cells. They help in the regeneration process in muscle repair after various damages. Here's cross-sectional area. How big are the muscle cells themselves? We could compare the bad outcome. Ostensibly, they're not likely to be in the placebo group versus the essential amino acid group. So again, this is some of the data that we could derive from the muscle biopsies. Here's a bit more. So we could also take those muscle biopsies, and here's Doug Foote in the lab, isolating some of those muscle stem cells so that we could grow them in cell culture. So basically we could take from our patients who are in one group versus another group, and also from young individuals, and then compare how those muscle stem cells grow and then react to different experiments. So this is a way of saying, okay, we have the human component, but then we can isolate these cells and then further analyze what's going on inside muscle cells and how they are or are not responsive to essential amino acids. Here's Austin again doing some analysis of some Western blots. So we could look at the size of various proteins. I don't need to go into details of that, but this is Western analysis. And again, all the undergraduate and graduate students coming through the lab get to do all this kind of work. We have real-time PCR, so you can think of messenger RNA. This is how we make proteins inside of our body. And here's just an example of a heavy amino acid that we can use as a tracer. So sometimes we infuse or we give by mouth tracer into individuals, and then we can measure how much of this or how little of this ends up in skeletal muscle, for example. That might be a little bit difficult to conceptualize, but I'm happy to take questions later on that. So a little bit further on the cell culture stuff. So we could take these cells and again, grow them under certain conditions. And then this will give us a better idea as to why some people respond well to the essential amino acids and some don't. So not everyone just has a good outcome per se, but we get more and more of this information will help us to identify how to prevent muscle atrophy in the long term. So that's this here. So again, maybe a biopsy from a bad outcome, and this could be before or after surgery, and to see if those the outcomes are different for the different experiments that we do in cell culture. And then we can also do this to young individuals who would have, in most cases, the best outcome under different experimental conditions, and to see how they are different than older adults. And I also teach a lot of this stuff in the classes that I teach. Here's a picture of John Meiskens and Max Ralph doing some stuff in the lab and having some fun. Here's Kate Kowalski. She and I were at the just outside the operating theater at Peace Health River Bend, getting ready to take on some muscle biopsy samples and then process it. This is usually like 6 to 8 o'clock in the morning on many days. Here, Chelsea Polisar doing some immunohistochemistry analysis. And I tend to talk a little bit fast, but I'll be happy to take some questions. Here's just a cross-section of skeletal muscle kind of blown up. Here's a nucleus. Here we have the cell kind of membrane. This is laminin. Each one of these are muscle cells, all of these. We could use this technique to look at capillaries, muscle cells, and nuclei that are inside and outside of muscle cells. We can also look at inflammatory cells, which seem to be a central feature of how people respond or don't respond to various treatment techniques. So I'm basically just filling time now, but if you guys have any questions, I'll be happy to take them.
We have some questions. Are these essential amino acids considered anabolic steroids?
Not steroids. No, steroids are different. So when I think of steroids, I usually think of like, say, testosterone or androgens, for example. But the anabolic term does apply to some of these essential amino acids. So we have 3 branched chain amino acids, leucine, isoleucine, and valine. And leucine in particular seems to have a very potent stimulatory effect on muscle protein metabolism. And one of the things that we could do is look at the signaling cascade. This is a complex signaling cascade of the anabolic response to various things, muscle cell, for example. This is a figure from a book that I use to teach seniors at the University of Oregon, as well as some entry-level graduate students. And we could look at mTORC1 pathway, for example, which is a very, it's like a signaling hub. So you can think, how do cells talk within themselves? How does the machinery get up and running? How are things kind of controlled at the cellular level? And here we have amino acids, and in particular, it would be leucine, that can have a potent anabolic effect on this signaling cascade, which can lead to, in this case, protein synthesis. Again, the amino acids are the bricks, proteins are the brick wall. So we can synthesize more of the brick wall, if you will. It's also involved in lipid or fat synthesis. So this is mostly occurring in liver cells, for example. Lysosome, autophagy, energy metabolism. So basic energy. So how can we actually move? How can we do work inside cells? Autophagy is a fancy term for the cell kind of breaking itself down. And then the lysosome is an organelle inside of cells that actually happens to be the storage locker, if you will, for amino acids. So that was a great question. It's anabolic, some of them, the leucine in particular, arginine and lysine as well, but it's not a steroid. So this is like something, this would be similar to if you were to take chicken or beef, for example, right? And you chop it all up and then you separate the essential amino acids from the non-essentials. We're giving them more of the essential because that seems to have a lot of really positive effects on muscle metabolism. And it's not just after surgery. It's, after maybe you're lifting weights, resistance exercise, you can have a more potent anabolic effect. People who are endurance trained, so people who run a lot, marathons, they tend to need more protein in their diets, for example. These are all components of some of the things that I do teach in my metabolism class. So that's a great question. Is there any other questions?
That was something I was going to just ask about. You're talking a lot about your lab and all the research that you do in your lab, and I was wondering what classes you teach as well. So if there's students that aren't necessarily in your lab studying.
Yeah. Well, so as you can imagine, I have a lot of students that work in the lab that want to go on to become orthopedic surgeons or physicians, physical therapists. I happen to be a physical therapist. I was a long time ago. And here is a picture that shows the class that I actually teach, 414, 514. This would be like a senior level. It's muscle metabolism. I teach other classes too, but it's mostly to graduate students. And a lot of what we just talked about, I do teach in this class. And since a lot of the students that are in human physiology want to go on to the health profession, not all of them, but many of them, the things that I do teach center around how does not having enough muscle have a negative impact on, say, recovery from surgery, recovery from hospitalization. You can think of the recent COVID effects and how that has had devastating effects on recovery of different patients. And I don't know if I have time to bring up a picture.
But we have one last question for you.
Okay, go ahead and ask the question.
The last question was, what can someone do to avoid getting osteoarthritis?
Exercise, eat right. The biggest contributor to osteoarthritis really is obesity. So if people are very large, they're carrying more weight through their joints. Let me see, I think I actually have. So this isn't a great image you can see here, but this right here is can be precipitated by joint angles. But a lot of this can actually just happen from excessive weight. So, and if you're not exercising a lot, the cartilage themselves, which is this picture here, the cartilage here, this cartilage here, actually does better if you use it more. So if you're not using it, there's a downregulation in the synthesis of cartilage, and that could lead to it being weaker, so that when you are putting weight on it, then it undergoes a little bit more damage per se. And with time, that accumulates, and it could lead to this bone-on-bone osteoarthritis, very, very, very painful knees. So yes, obesity is the leading cause. So if, again, exercise is the main component to, or lack of exercise really is to that. It's also diet contributes, but the main, I mean, you could basically be on a McDonald's diet and exercise like every day, and you could attenuate or reduce the bad effects of the McDonald's diet, for example. Really, the things about McDonald's diet is not so much fat per se, but it's the sugar and refined sugars that are in it. We talk about this in my class, but Phil Matern, another faculty member in the department, has a metabolism and nutrition class, goes over the fundamentals of this sort of stuff as well. So I think our department is geared very nicely towards kind of like helping to understand all these things. And so if people are interested, students are interested in the health profession, there's a whole number of things that could be. A lot of the work that we do also kind of involves biomechanics, which Dr. Karduna will talk about a little bit later on.
Well, that's a great segue right there, because he is up next. But Hans, thank you so much for all of your slides and information.
My pleasure.
Yes, you took us deep into the cells. So let's see where Andy Karduna takes us. You are next.
Can you see my slides?
I can see your screen.
We see your
Hold on.
file folders.
Oh,
there you go.
Okay, good. Okay, thanks. So yeah, so as Hans was saying, you know, a lot of the faculty in our department, we focus on issues related to the human body and health, and we all have our different perspectives. My background is in bioengineering, and specifically, I'm really interested in mechanics. And so the combination of looking at like the forces, things you might focus on in physics, combination with physiology and biology is referred to as biomechanics. And specifically, I look at biomechanics of what's going on in your upper extremity, specifically, I'm going to talk about a study that I do focusing on the shoulder. So when people think about the biomechanics of the shoulder, this might be something that you look at. You know, all the muscles, the muscles that Hans was talking about, that Ian talked about, that drive the motion. The shoulder muscles are really big and complicated. And this is just a general overview of what they might look like. But as Ian talked about, the muscles don't move themselves, right? The muscle is driven by a signal from the brain. And so you can think about how we move, and the forces are a combination of the kind of what's there, what muscles are there, but then how the brain drives those muscles. And so in my lab, I'm interested in studying both what happens locally at the shoulder, because like Hans, I'm interested in working in patients who have orthopedic injuries instead of at the knee, injuries at the shoulder. But I'm also interested in how motion is controlled at the upper extremity, because just sitting here doing this, like I'm moving my hands around, I need the muscles, but I also need my brain to control those muscles. And so one of the areas that I am interested in studying is a phenomenon called proprioception. So that's a sense of where your body is in space. So the way we know where we are in space, so I have a little picture of a little kid, so you can use your vision, you have some sensors in your inner ear, your vestibular system, but then you have your proprioceptive signal, which is signals coming from the muscles. So the best way, so I can't see anyone, but I'm gonna ask you to do this very simple experiment. So other than Hans and Ian both told you to think about but not move, I'm gonna actually tell you to actually move. So I want you to do this. Try and close your eyes and kind of see what the subject on the right is doing. So if you kind of hold your hand up like this, close your eyes, and then touch your nose, okay? So if you try that, you can see Dr. Dreyer doing it very well there. Somehow you were able to know where your hand is in space without looking at it, right? So you can imagine looking at it, but somehow if I close my eyes, I still know where my hand is in space. So the sense of where our body is very important. I'm standing right now. Somehow I'm able to stand. I'm not looking at my feet. So I need to know where my body is in space in order to help control my movement. So a lot of the experiments that I do in my lab are to better understand this sense of proprioception. And for a long time, I was doing that with a combination of just regular technology. I would put motion sensors on. We developed an app for a phone. But more recently, I'm interested in integrating my research with virtual reality. So some of you might have seen virtual reality technology. You can use a phone. There are products that you can buy that are actually not that expensive compared to what they were, say, 10, 15 years ago. Some of you may have seen them and played games on them. We're less interested in the games and more interested in how we can integrate this technology to use it to better understand how we move around in space. So what I thought I would do is just walk you through a simple experiment that one of my graduate students and undergraduate students, so the graduate student's named Kate and my undergraduate student Kylie, worked on this project together. And kind of like what Hans had described, we have students in our lab. We have graduate students, people getting their master's or PhD, but we also have undergraduate students. So if you were to come to the University of Oregon and become a human physiology major, one of the opportunities that you might have is to work in a lab in the department. So what we did is we created a virtual reality space. So it's very boring space. Like you can create a lot of really complicated things in virtual reality. This is probably the most boring virtual reality space that you can think of. It's a couple of walls and a ball. And the ball represents where your hand is in space. We didn't want to provide a lot more cues. So I'm going to kind of walk you through, and again, this is typical of the kinds of experiments that we do in my lab. So what we asked people to do, you can see the cartoon on the left, is we had subjects basically lift their arm, lower it, and then try and replicate it. So like a good example would be if I go here and I replicate it, that'd be great. But if I go here, and then when I replicate it, I go up here, that's bad, or there's an error. And we're trying to see the difference between the first try and the second try. And we've done this in the past with people's eyes open, and we've done it with people's eyes closed, okay? And until we integrated the virtual reality part of it, those were our only two options. eyes closed and eyes open. So for this particular protocol, what we did, you can see this is Kylie with this is the HTC Vive virtual reality goggles. You can buy them on Amazon. So she's seeing that really boring room with the ball, and hopefully this video will play. Let's see. It's like 50-50 there. So you can see the ball, that just represents this graph as she moves her hand up, right? This graph just represents the angle. So that's the target. So she's at maybe, say, 70 degrees. She lowers, and then she's going to replicate that task. Okay, and in this example, you can see that where she started was about where she ended, but we can look at the difference. So we can get the error, the difference between the first time she was there and the second time she was there. And if she's really good, that error will be very small. And again, over the years, we've done this under just those conditions, either eyes open or eyes closed. But with virtual reality, we can really manipulate. So what we did with this experiment is we had them with vision, so accurate vision and no vision. And that's what we've been doing. But the great thing about virtual reality is we can offset their vision. So what Kylie would see in that is her hand would be here, but that really boring virtual reality could have the ball here, or the ball could be here. So basically, we are trying to trick her into thinking that her hand is in a completely different place. And then, remember I said there's multiple ways I can know where my hand is. I can either look at it, or I have the proprioceptive information. And generally speaking, those are aligned. I can use either. I can either use vision or proprioception. And we want to know which one of those things do people rely on more. And it's a hard experiment because if you have both, you don't know. So what we do is we manipulate the experiment to create an offset. And then the question is, do people care more about what they see or what they feel? And this is important because A, it helps with understanding movement, but it also helps in patients that have either vision problems or proprioceptive problems or some integration problems in their brain, which prevent them from taking that information correctly. So can they rely more on one sense than the other? And what we found in this experiment, and so I'm just kind of walk you through, I just have one graph here. So on the y-axis here is the error. It says constant error, it's just the error, right? And so I said there were three conditions. So if you look at the ones on the left, the kind of gray and the one next to it, those are accurate vision and no vision. So if we give Kylie accurate vision and then we take away her vision, it doesn't seem to change. She's maybe off by maybe 2 degrees or so. But when we offset her vision by about 8 degrees, she's off by 8 degrees. In this experiment, what we found is that people care more about their vision than their proprioception under these conditions, right? So she could have used either. She could have used proprioception or she could have used vision. So this is a way we can utilize the technology of virtual reality to kind of manipulate people's environment. And again, this was a very, very simple experiment, but our plan is to build on this and look at lots of other patient populations. We can look at athletes. We can look at the kinds of like the Hans talked about patients with that have total knee arthroplasty. We could do the same kind of thing on those kind of patients. So that's, again, just a summary of the kind of stuff. And I just want to walk you through the kinds of experiments that you might see in a lab in our department. So I'm happy to take any questions that might come up, Laura.
Yeah, so there is a question about, do you ever do research with people who are visually impaired, and how do you handle that?
Yeah, so I mean, generally for this experiment, we are working with people that either have, either 20/20 vision or corrected to 20/20 vision. So they will either have glasses or contact lenses, so that we're not worried about that being a factor. However, one of the things that I'm interested in doing is actually looking at people with impaired vision, because I can impair their vision like I just did for Kylie here, we can create an offset, but we can also look to see, are people who have poor vision in the first place, right? Do they weight their proprioception more? So I just gave you an example where in this case, vision seemed to be really important, but if someone has an injury, maybe they have a cataract or glaucoma, in those individuals, do they rely less on vision and more on proprioception? That's a great question. It's just we have not looked at those patients, but that's something we've been talking about doing.
So Andy, what is the next, like who takes your research? What's the next step with your research? Like other research is immediately goes to teaching new surgeons, like Hans was talking about. What are your people you're teaching going to do next?
So you mean my students?
Your students and I guess your research. I guess maybe it's two questions.
So the students would be similar to kind of what Hans is talking about. A lot of our students are interested in the medical field, especially at the undergraduate level. They're interested in becoming physical therapists. I've had a lot of students go on to medical school, physical therapy, OT, and the like. In terms of my research, a lot of this particular research, I also work with surgeons, but this particular one is more applicable in the physical therapy realm. So there's a physical therapist in town that's playing around with using virtual reality to encourage people to exercise. So he's got a virtual reality system where you can imagine if you're exercising like this, it can get kind of boring, but he has an environment where you put on the virtual reality goggles and it's kind of like a popping balloon thing where you're playing games where you're trying to pop balloons. So all of a sudden it becomes a contest and you're trying to get points as a way to encourage people to move more. And there are other researchers in the world that have done this with people with low back pain and found that if you do that, they're going to move more. And by moving more, that's going to encourage them to promote the healing process. So this has a lot of applications in the physical therapy realm.
Fantastic. We have one more question and then we'll pop over to Andy Levering. What other examples where you have used the VR technology in research?
So right now, this is my first attempt with using virtual reality. Like I said, I've been doing these kinds of experiments for a long time, but it's just in the past maybe 2 or 3 years that I've started playing around with virtual reality. There's a lot of potential applications. I talked about looking at rehab as one example. There are also lots of applications in terms of there are probably maybe 5 or 6 labs on campus that are interested in using virtual reality. Everything from the biomedical interest that I have, they're looking at it in psychology. Virtual reality can be used for everything from like you can present someone with an avatar that looks like themselves or that doesn't look like them. You could change someone's skin color. You can change someone's gender to try and get them to better understand the different environment. And then there's a bunch of people in the School of Journalism and Communication that are using it as a way of like maybe the next stage of communication. And again, we're using the same technology, but for vastly different applications.
That's awesome. Very, very impressive. Well, thank you so much for sharing, Dr. Karduna. We will move on to Andrew Levering next and circle back if there's any additional questions for you all. But Andrew Levering, Dr. Levering, welcome.
Thanks so much, Laura. Thanks to everybody for joining today and all the great presentations. So I'm going to try to follow the examples of my colleagues by having you sit there and imagine doing something and then doing it. So I want you to imagine going to one of these wonderful places here I have in the pictures, because it's not going to happen right now thanks to COVID restrictions. So you can only imagine it right now. There's no way you can travel there. So I'm an environmental physiologist, and what that means is if you were to ask my colleagues, what do I do? They would show you pictures like this and tell you that I go to places and play in the mountains, play in the snow on top of the mountains here, or go scuba diving in places like Croatia, or play with or interact with astronauts on the space station, because that's the kind of research that we do. However, what I actually do is I just pack up everything in my lab into Pelican cases and then ship them all over to places all over the world, take it all apart, rebuild my lab, and end up working inside some lab somewhere in just a different location. So for example, these studies that were done in Bolivia that I worked with Dr. Dreyer on the mountain in these studies as well. This is what the actual laboratory looked like inside. So this is what we could see outside, but this is what it looked like on the inside. So we're just doing the same kind of research we would do in our normal labs, just in a different location. Same thing here. This is on a Croatian Navy base in Split, Croatia. So here's some scuba divers going out to do a dive. And then when they come back into the lab, we take some of their blood, we make some ultrasound measures of blood flow in their arms and things in their heart and lungs to see how scuba diving impacts them when they go out onto a dive. And then in space, we're worried about how sleep disturbances can impact visual problems in space. And so because we can't fit everybody onto the space station to do these studies, it's easier to do what they call bed rest studies. And so these bed rest studies, you see all of these subjects, they're on a bed that's tilted down about 6 degrees, so all the blood flows to their head, kind of like how you would feel if you were standing on your head anywhere. And this is a way to simulate low gravity conditions or microgravity conditions that they have on the space station, because when you're in space, you don't have gravity pulling your blood down to your lower extremities. It actually all ends up in the head. And they think this might be contributing to these visual impairment problems people have in space. So over the last eight or so years, I've been traveling to different countries, different states, different places to do these types of studies for altitude diving, if it's scuba diving or if it's apnea diving. And then for the space studies, they've all been done either in Germany or upcoming ones going to be done in Houston. And some of these some of these dates have been shifted thanks to COVID. And I just haven't updated this, so I apologize. The part of physiology that I'm interested in studying in these different areas across the globe are cardiopulmonary or heart and lung issues. And so one of the big things that we like to study in my lab is a small hole in the heart that's present in about 1/3 of the population, meaning that every third person that you were to count, they probably have this small hole in their heart, and most people don't even know that it's there. So if you're looking at the cross section of the heart here, we have the right atrium, the right ventricle, the left atrium, and the left ventricle here. And you can see this kind of square here blown up. 1/3 of the population has a little hole here that allows blood to go back and forth across that hole. And we found that surprisingly, that the presence of this little hole in your heart can cause some significant differences in the physiology that we measure. And so in my lab, if we want to know if somebody has a hole in their heart, we don't have to have a fancy multi-million-dollar MRI machine. We have a fancy less than $1 million ultrasound machine. And this is a picture of the ultrasound image of the heart. So if you can imagine, I'll show you. Is it gonna flip for me? There we go. So if you imagine when you image it with an ultrasound, you basically turn the heart upside down. So now the right ventricle and the right atrium are on the left side of the picture, and the left ventricle and the left atrium are on the right side of the picture. And if we want to know if there's a hole in the heart, we inject small air bubbles into the blood. I know that sounds dangerous, but it's a routine clinical procedure that they do to detect this hole. And I'll show you what happens when we inject these bubbles. So you see the heart beating here, and you'll see some bubbles appear, and then you see them jump right across from the right side to the left side. And I'll show that video again so you can see that whenever it gets to the end here. Let's see if I can play it again. Okay. So again, this is the right side of the heart. We inject these bubbles into a vein. They'll travel to the right side, and then they'll jump right across that hole into the left side. And so we know this person has a small hole in their heart. In this case, this person has a relatively large hole in their heart because that's a lot of bubbles getting across in this individual. And so we know a lot of people in this town that have this hole in their heart because we screen pretty much anybody that walks into the lab for the presence of this small hole. And I will jump to some of the research that we've done in this area on this small hole, which is called the patent foraminal valley, or PFO. And so one of the initial studies that we did, and these pictures are of the undergrads that have worked on these projects in our lab. So this is Sierra Hill and Amber Ng. Sierra is now a physical therapist here in Eugene, and Amber is finishing up medical school in Lebanon, Oregon at the DO school. And one of the things that we initially discovered about this hole in the heart is that if you have this hole in your heart, you have a higher body temperature than somebody without. And so this figure here in the red open circle, so they're open circles because there's a hole in their heart. So there's also a hole in the symbol here. And they're red because they're slightly warmer than the ones in the blue. So these are the people that have the higher temperature. So we have temperature here, and then we have pre-exercise, and then several intensities of exercise. And they start off warmer, they stay warmer, and they're always warmer all the way through maximal exercise. Now, if you're looking at the numbers here, you think, oh, that's only about 3 or 4 tenths of a degree C, 3 or 4 tenths of a degree Celsius difference in core body temperature. But if I was to measure your temperature right now and increase it by 2 tenths of a degree, you would start to sweat because you would want to lower your body temperature back down. So these people are about twice that sweating threshold different from another human, and they're not sweating when they're at rest. So they're not trying to lower their temperature. They like that higher temperature, and they like to keep that temperature higher. When we went to do some of our altitude studies, we noticed that the people that had this hole in their heart, all of them got sick on the first day. So if you have this hole in your heart, it increases your risk of developing mountain sickness. And we also discovered after keeping them on the mountain for several weeks that they didn't change their breathing the way they were supposed to, or the way people had measured in the past. And so we did some more studies in the lab here. This is a picture of Madeline Hay. She was the undergrad who worked on this project. And Maddie is also now working on her DO at the school in Lebanon. And what we see here, again, the red subjects are the ones that have the hole in their heart, and the blue subjects are the ones that don't have the hole in their heart. And what we did in this study is we put these individuals in a hot tub to increase their body temperature, because it's well known that if you increase somebody's body temperature, they'll start to breathe more. So increasing somebody's core body temperature is a great way to increase their breathing. And what we found is in the people that don't have a hole in their heart, as their core body temperature increased, their breathing started to increase. And so they got to this threshold break point, and then they really started breathing. So then they really start to hyperventilate. Well, in the people who have a hole in their heart, we got them about a degree C warmer before they started to breathe. And then when they started to breathe, they really didn't breathe a whole lot. So they have blunted ventilatory responses to things that you would not expect people to have blunted ventilatory responses to. And unfortunately, I have no idea why a hole in the heart would impact somebody's breathing responses, because technically the things that drive breathing are located in the brain and not in the heart. So there's some connection between this small hole in the heart and the brain, which is where we determine if we're going to breathe and how much we're going to breathe. And then just to follow up on those studies, we did another thing that it's well known that will increase breathing, and that's having somebody breathe carbon dioxide. So that's CO2. So here are the undergrads that worked on that project. This is Alex and this is Alyssa, both in medical school right now. Alyssa's at Michigan and Alex is at OHSU in Portland. And here we have in the dark line here, dark solid line, these are the people without the hole in their heart. And here in the dotted gray line, these are the people with the hole in their heart. And as we increase carbon dioxide levels in their blood, you see that people normally increase their breathing in a very linear fashion. But if you have a hole in your heart, you don't increase your breathing as much. So this was the third example that we've become aware of, where if we challenge somebody's breathing, they just don't breathe as much as the other group. And again, we have no idea how a small hole in the heart is communicating with the brain to have these differences in their responses. So the ongoing studies in the lab for certain, trying to figure out why this small hole in the heart causes these physiological problems. Now, this is just the physiology side of things. So this is just Andy being interested in breathing and wanting to go to Croatia and the mountains in Germany during Oktoberfest and things like this. So there are also clinical reasons to study the PFO. So I'm going to show you a video and try to navigate while we're doing this video. It's put together by St. Jude Medical. So one of the downsides to having this hole in your heart is that it allows blood clots that would normally go to your heart. So you see these are blood clots here coming into the heart. Normally these blood clots would go into the lungs and get filtered out and not cause any problem. But if you have a hole in your heart, these blood clots can sneak through. And when they do that, they get over into the left side of the heart where they get pumped out into the aorta. And once they get pumped out into the aorta, they can go anywhere in the body. Unfortunately, if they go to the brain and block an area of blood flow to the brain, it can cause a stroke. And so people are really interested in this PFO. And because of that, there's a device, several devices actually, that they can put in. Here's a catheter image where they put this device in and they can close that hole in the heart so that and you can see here the other side of the catheter. And then they disconnect this device and then it's there permanently inside the person's heart. And after several months, the cells of the heart will grow over it and they call it endothelialization. So the next time a clot comes, it hits that, it's not allowed to go across the heart, it goes to the lungs, and therefore it prevents the person from having a stroke or any kind of issue downstream. And other studies that we're doing right now with the local cardiology group is identifying these people that are gonna get that hole closed in their heart. And we bring them into the lab and we study them, measuring everything that we know to study right now before they get the hole in their heart closed and after they get the hole in their heart closed. So, which is great for us because these people are getting something that's going to help them not have a stroke downstream, but it's also really good for us because it gives us a lot of insight into the physiology to determine, is it really that hole that's causing this problem, or is it just something related to that hole? So if what we're measuring doesn't change after we close the hole, then it's probably not the hole that's causing the problem. So that's all I got. Those are my.
That is amazing. Oh my goodness. We got to go around the world with you. Well, we are close to our time, but we do have some questions. So if you all don't mind to hang on a couple minutes, we'll go through these couple questions. But for you, Dr. Levering, somebody was just curious, what causes this hole? Do we know why people have this hole?
Yeah, that's a good question. So the short answer is everybody has that hole in their heart when they're still a fetus inside their mother, because there's no need for blood to go through the lungs when you're inside your mom. Your mom is doing all the breathing for you. So you don't need to have blood flow going through the lungs. And so having this hole in your heart when you're a baby allows the blood that comes into the right side of your heart to just go straight to the left side and downstream where it needs to go, primarily the brain in the developing baby. So it's just that for some reason, 1/3 of the population, that hole doesn't close after you're born. And nobody really knows why that hole doesn't close. And I would tell you, before I started down this path, most people would tell me or tell you that, oh, that hole doesn't really have any functional consequences or physiological consequences. And pretty much everything I measure is different in these individuals that have that hole or don't have that hole.
Well, somebody tacked on to that was just asking if this hole plays a positive or negative role in the level of physical activity. So you've kind of talked about that.
Yeah, so there is an idea that having this hole in your heart will impair your ability to do some types of physical activity. It's pretty challenging. It's a challenging question to study because you need to find people that are relatively well matched for other things that contribute to physical performance, like muscle mass, training status, age, biological sex, and things like this. So those studies haven't been done well. So you really need to match things. There's so much more to performance than just the small hole in your heart. That I don't think it's going to be what makes or breaks anybody's performance. Now that said, if two people are very well matched and one person has an extremely large hole in their heart, like some of the people we've studied, there would be some potential issues in their ability to perform as well as somebody without it.
What about does this hole correlate with dying at an earlier age? There's a lot of concerns about this hole right now.
Yeah, I know, because 1 in 3 people have it, right? And then you don't want to believe that you're one of those 3. I don't have it, but that's only because I've been screened and studied so many times in the lab. So there's, as far as anybody's ever reported, there's no reason to believe that this is going to cause you to die at an earlier age than somebody without. And there's a really good study of about 1,000 autopsies where they looked at the prevalence of this hole in people going from age 1 to 99. And the only thing that came out of that age-related study, and again, it's an autopsy, autopsy specimen, so it's not the general population, but that the prevalence decreased in older people, and in the older autopsies that they looked at, but the size of the hole was larger in the older people. So it could be that there's some beneficial effect of there's something beneficial about this hole, but nobody nobody knows for sure. Nobody knows for sure.
Well, fantastic. Thank you for all of your research and sharing with us. There's a few questions left, but we are out of time. But I do want to wrap up with the last. I really would love, there's a student who is curious if there are any volunteer opportunities ever in any of your labs, and ways to connect with you all if this is, they're excited. So.
Yes, absolutely. In my lab, I have right now 15 undergrads working in my lab. So
definitely multiple opportunities to volunteer in the lab. And also during the summer, we try to find ways of paying and compensating our students for their work in the lab as well. So this is also, can be thought of as a job. I mean, your experiences as an undergraduate may actually determine their career path in the future, and doing this type of research shouldn't be just thought of as a volunteer opportunity, but also as a potential career. So definitely start thinking about that now.
And I would say that by saying that this is how I got into doing what I'm doing now. When I was an undergrad, I volunteered to work in a neurophysiology lab, and that's why I do what I do now. It really changed my life.
I just add that I shared my email in the chat, so if that's somehow communicated to the rest, feel free for any students to email one of us. Or there's also online, you can just search us, Human Physiology at the U of O websites. So
yeah, and I
believe echo everything that everybody else said.
Each of our labs has a website as well that you should feel free to explore, and reach out to us and our students just to find out more about what their roles are like if you are considering doing research.
Well, thank you all so very much for taking this time. I know this is a big deal to carve out in your busy research worlds. And for everyone watching and for our researchers, this presentation has been recorded and it will be posted on our website next to this talk. If you want to look at it again or share it with anyone else, please do. We always make resources available to our viewers as well. So if for some reason you can't find their labs, you can always email the sales staff at uoregon.edu and we will help connect you. So again, Physiology Department, you are fabulous. Thank you for taking the time, and we will be talking to you soon.
Thank you so much, Lara.
Take care, all. Thanks a lot.
Bye, everyone.
Take care, everyone. Take care. Have a good day.
Okay.